Hello, and thank you for this invitation to give a presentation, a keynote presentation for Curating the Clinical Genome Conference. I've chosen as my title, Curating the Variome, in honor of my dear friend Dick Cotton, who died in 2015, who launched the Human Variome Project as an attempt to pull together international efforts to understand and share knowledge of variation in the genome. My chair, Helen, will see herself in this picture, and actually this was a gathering I organised 10 years ago, almost exactly, at Hinxton, entitled Collaborative Curation of the Variome, because at that time we had a programme funding from our government to try and pull together genetics and genomics research in the country. And actually that's Dick at the back of that group there, but in fact, to be honest, we had to Photoshop him in because he was so busy rushing around the campus trying to get support for the Human Variome Project, which was his endless effort prior to his demise. I thought we might just begin, not 10 years ago, but 40 years ago, when I was working at Great Ormond Street in training. We were given the first opportunity to do a molecular diagnostic test, restriction enzyme cutting of the variant that caused sickle cell in beta globin. And this, of course, is still one of the most important variations in our genome in the world. About 250,000 children develop and many die of this condition every year. And about 1 in 3 of the people in the Rift Valley are carriers of this because of resistance to malaria. And indeed, it is as a result of the ethnic minorities in this country, the commonest recessive disease that we face. At that time, life was very simple. We could literally list on one hand the number of molecular diagnostic tests we could do, but it rapidly got a lot bigger. And of course, in those times, in the 80s and the 90s, our primary resource was Victor McCusick's Mendelian Inheritance in Man, which in 1985 became OMIM, and I was pleased to be a part of the editorial committee for several years in the 1990s. And that was our single textbook or online resource for variant interpretation. But of course, it got rapidly a much bigger challenge. And in 1997, UNESCO made a formal declaration that the benefits from biology, genetics, and medicine concerning the human genome should be made available to all with due regard to the dignity and human rights of each individual. And it was this which stimulated Dick to try and stimulate our group, our community, to think about how best to share that information. And so he launched the Human Variome Project in 2006 formally, and Johan Den Dunnen was a key player, having put together the software in Leiden, the LOVD software, which is a free resource for anyone to set up databases of variants in their genes of interest. And indeed is still going strong. It receives a feed from the Netherlands geneticists and also from around the world, those who are interested in different genes. And apparently he has about 1 billion variants now using their service, so really a very important resource. The idea, as I said, was to create an online reference database using these tools so that we could all access information wherever it was derived from. And indeed, it did very well initially and became a recognized non-governmental organization at UNESCO and reached its zenith in 2012 when we had what now looks a rather strange picture in these days of social distancing, where we all gathered with David Weatherall as our keynote speaker. But the problem was and remains, how do we resource these databases and how do we get people to share their data and make it available to all? Because it was very clear that as with our friend from Lord of the Rings, people can be a little possessive and hang on to their data and not share it. Sometimes for very good reason because of the confidentiality issues, but as often as not, it's just that they don't want to give away that which has spent so much time gathering. And so trying to make it possible for people to share but still be given credit for their work is one of the challenges we did face and continue to face. One of the most successful databases that came out of the LOVD system, and indeed which predated the Human Variome Project's formal launch, was that of our Insight database. We formed Insight in the early 2000s, merging together the FAP and Lynch syndrome research groups. And as its first chair, I asked the 3 teams in Finland, Newfoundland and Holland, who were running databases, to come together in a workshop, and they agreed to form the Insight database, which has now become an international reference standard, and we hope will soon be incorporated into the ClinGen VSEP system. It's run out of Melbourne and gathers data on all of the colorectal cancer genes, and is a good example of what I hope will become standard for all disease groups in the years to come. Of course, the world changed quite significantly with the emergence of whole genome sequencing, next generation sequencing, and the Global Alliance for Genomics and Health was the next major player in the space, brought together by the Broad Institute, the Science Centre and the Ontario Cancer Institute as its primary sponsors, along with the European Bioinformatics Institute at Hingston. And the idea here was to try and pool resources from the people now generating vast quantities of data through genomic sequencing. Ewan Burney is now its chair, who leads the EBI, and Peter Goodhand, who is a Yorkshireman based in Canada, is its primary organisational leader, and I'm very grateful to him for these slides, and Peter and I are good friends. So the GA4GH, as it's now known, has really continued to flower and blossom, and created a substantial ecosystem in multiple countries with large numbers of subscribers, 24 driver projects now, and it's very much focused on technical standards and regulatory processes and policies. So this has been a great success in pulling together our global efforts. And when it was launched in 2014, we were looking for driver projects, and there were now a very large number of them. Initially, there were 2, Matchmaker Exchange and the BRCA Challenge, and I was involved in setting up the BRCA Challenge, or suggesting that as a way of bringing together the traditional genetic approaches that were feeding into LOVD and other databases, and the new genomic approach, to try and pool our knowledge around the 2 genes which Angelina Jolie had made so famous, BRCA1 and BRCA2. In fact, it was very much in my thoughts at the time of that meeting in 2014, as I was trying to sort out this family, which the clinical geneticists and clinicians in the room will understand the challenge of. Here were 2 sisters who'd been investigated by a colleague. Both had been shown to have this missense variant. One of them had early onset breast cancer, and they had initially been told that it was a variant of uncertain significance, only for their cousin to then develop an ovarian cancer, which was metastatic with the same variant. And so we're asked, is this or is this not a pathogenic variant? And the interesting question, the interesting reason for this is because the Bayesian probability calculation had changed quite dramatically in those 7 years. The reason it had been judged to be a variant of uncertain significance was a case of co-occurrence. This variant was seen in a single patient in America who also had a variant in BRCA1 in the other allele. And since it was considered not viable to have 2 faulty copies of BRCA1, Doug Easton and colleagues in 2007 judged it to be a variant that was probably not pathogenic. However, when Susan Domchek found that patient, it turned out that she was short, had microcephaly, was very drug sensitive. And so she was the first example of someone developing what we call Fanconi's anemia with biallelic BRCA1 involvement. And so what we'd effectively done was cancelled out the co-occurrence in the Bayesian calculation. And when you looked at the rest of the evidence, it was actually very much in favor of this variant being pathogenic, albeit probably hypomorphic to allow the patient to have survived with 2 faulty copies of the gene. And indeed, when we went around the country and asked people for an opinion on this family, pretending to be a member of the family, we found multiple different interpretations of the pathogenicity. So clearly, we needed to pool our knowledge and all be telling the same story. The important thing in these Bayesian calculations is always to remember that evidence can swing you both ways. And so it's possible to be changed from something that is to something that is not a variant of importance. So the idea of putting together our efforts was well received at the launch meeting, and Stephen Channock, shown there, who is the head of the Genetics and Epidemiology Unit at NCI, and I were asked to pull together a group of experts in the space, and we all gathered at the UNESCO headquarters initially in Paris, and set off with the BRCA Challenge, and Heidi Ream designed our logo for us. And it's been very successful. It's now led by Melissa Klein as our coordinator in California. And in 2019, we were able to publish our first report. We succeeded in producing an impressive database, which now has more than 40,000 deduplicated variants. When we started, when we pulled together the BIC data, the Clinvar data, the LOVD data, and the universal database from Paris, there were fewer than 6,000 variants available, and not all of the center teams had those in their databases. So this has demonstrated just how much data is out there if only we can gather it together into one place. I suggested it would be really good if we could have an app for this, and Gunnar Rasch and Faisal Al-Kadumi in Zurich succeeded in producing for us the BRCA Exchange app. So when we have cases like the one I gave as an example, you can actually flag the variant you're interested in and then be notified if that variant changes from variant of uncertain significance into pathogenic or non-pathogenic. The analysis of these variants is carried out by Enigma, which was an established international group led by Fergus Crouch in the States and Mandy Spurdle in Australia. And that group continues to prosper and will hopefully soon be recognized into the ClinGen VCEP system. So ClinGen, of course, set up by NIH alongside Clinvar, and Clinvar, led by Heidi Ream, has really done very well in pulling in data from, especially from the American laboratories. Although one of the limitations of Clinvar is you need to enter the data individually, and we're still working to try and share data between the LOVD databases and the Clinvar database. But hopefully, as this system continues to mature, our interpretation and clinical access to variants will improve considerably. And of course, the other thing that's really made a huge difference was the work of Dan MacArthur and colleagues, now led by Heidi and Mark Daly at the Broad, pulling together now a very impressive gathering of 76,000 genome samples, so that we've got some baseline data to compare to. And the exciting thing is that this latest update contains a lot of other ethnicities than the North Europeans, which made up the bulk of the database initially. So we're starting to see progress in pooling data, and I should also perhaps mention the variant arc at Genomics England is another example of that. But understanding which variants actually are pathogenic does require quite a lot of effort and pooling of knowledge, both clinical and functional tests. And the functional tests are now starting to make a big impact, especially in the cancer genes, where I have my greatest interest. So this is a picture of Leah Starita, who works with Jay Shendur, and Greg Finley did his PhD on this very nice work, which has been incorporated into the BRCA Exchange. The idea here was to actually produce each potential variant across the areas of interest in BRCA1 in a haplo-dependent cell line to establish whether or not a particular change would cause the gene to become non-functional, and cell death implying loss of function. And that allows a functional score to be given to each missense and each variant and incorporated into the Bayesian calculation, along with the segregation and other data available. In the case of the mismatch repair genes that we work on in the InSight database, of course, this is a particularly easy system in a way, because this is a well-established pathway that one can challenge in cell function. And a whole cluster of new mismatch repair gene analyses have been developed with these 3 publications in the last couple of years, stressing the system and working out which variants in the key genes in the mismatch repair system are pathogenic. So I think functional assays are going to continue to evolve as the technologies allow. And of course, in the case of mismatch repair, we have 2 well-established functional assays in immunohistochemistry and MSI testing. Immunohistochemistry, for those not familiar with this area, looks at the 4 proteins produced by the 4 key genes in the mismatch repair system that cause Lynch syndrome. And the Promega is the standard assay for microsatellite instability testing, looking for slippage in the very large mononucleotide repeats which are used in this assay. The problem with that is that those repeats are so big you can't use them reliably using a next-gen approach. The stutter peaks make their interpretation challenging. So the top 2 traces are normal, the bottom 3 are abnormal, but it requires a trained eye to be sure which ones are and which ones are not pathogenic. So a few years ago, when we were moving into this space, my colleagues and I set out to try and produce a new assay which would be amenable to next-gen sequencing as a functional assay. And my colleagues Mike Jackson, Maura Santibanath-Koref, as the leads of this, along with our PhD students, produced a very nice assay which has now been published. And the idea is to take short repeats linked to a polymorphism. The polymorphism, when analysed using a molecular inversion probe approach, allows you to not only amplify the repeat, but also the linked polymorphism, allowing you to work out which allele has changed in length. And building in a barcode allows you to do a molecular count. And then when doing the bioinformatics, you can separate the noise of PCR stutter, where both alleles will be equally represented, from the signal where only one or other allele will have shown slippage, as these shorter repeats don't slip quite so often. And using 24 of these markers, and indeed only 6 of the best ones, it's possible to separate the microsatellite high from the microsatellite stable cancers. So this allows an extra level of information to be fed into the sequencing data, where we've got an MSI high tumor, and furthermore you can use BRAF testing, as about half of the sporadics will show this variant. So a single assay can give you the information you need to sort out which ones are likely to be Lynch syndrome, and then move on to germline testing with that additional information from the functional test. What about phenotype? So phenotype-genotype correlation is of course critical if we are to reliably identify clinically significant variants. Decipher, the product of Helen and colleagues in the Sanger Centre at Hinxton, is a classic example of where we've been able, or they have been able, to pull together variant information with detailed phenotypic information from the partner clinicians involved in the process. And by pooling that data and using ever more sophisticated analysis, it's possible to reliably group together the right patients and interpret the variants more reliably. In the case of Lynch syndrome, we have established a prospective Lynch syndrome database designed by Paul Muller. And what this does is gather data from more than 6,000 patients with Lynch syndrome, followed prospectively from when they have their first colonoscopy at the age of 25, to see who gets what cancer and when. And what that's demonstrated is that with different gene variants and different genes involved, you get different patterns of cancer. So we're able to actually attach a more reliable phenotype to different variants, and also understand that in fact a proportion of these patients don't develop cancer, which is obviously important in doing the calculations of penetrance in families. Looking more widely, in Britain we have the benefit of having a National Health Service and having an integrated healthcare record. And about 10, 12 years ago, Jem Rashbas and I were put together on a committee at the Department of Health to think about how we responded to European directives on caring for rare disease patients. And we realized that we could potentially draw together 2 strands of information using technology that Jem and his colleagues had developed. So the National Disease Registration Service, which now embraces the National Cancer Registration Advisory Service, which we were then talking about, is based on the NHS records. And it has, in British law, legal permission to collect information about patients, granted under what's called Section 251 of the Health and Social Care Act. And of course, this is a very sensitive issue, so it is under review annually by the Confidential Advisory Group of the Health Research Authority. And patients can opt out of it, although this is very rarely the case, because people have a trust in the NHS in Britain and are happy for their data to be used in this way. Importantly, the data is aggregated and can be anonymized and depersonalized wherever possible. And what Jem explained to me was that we could use that in relation to our NHS genomic laboratories, using what they call SALT. the 256 hash code, which basically takes the patient's ID, NHS number, and date of birth, etc., and mixes it with 256 bits of random information to produce what's effectively an uncrackable code. So the idea is that we then would go to our NHS laboratories and take the data from the NHS lab on NHS number and date of birth, put it through that cryptographic hash function, making it an impenetrable code, and use exactly the same process at the cancer registry to produce the same unique pseudo ID. And this data could be shared across the genome, across the internet, without fear of patients' identity being released. The demographic data, which is not personal, doesn't require quite such a level of anonymization. So this is a reversible encryption, still allowing it to be transmitted across the internet. And then finally, the genetic information from the labs could be shared in its true form and married together with the unique pseudo ID. So that in essence, what we could do was take patients who'd been identified as having what we thought was a pathogenic variant in one of the key genes, initially the cancer genes, and then look to see whether in fact they got cancer down the line, and in that case, when they got it in what tissues. So we set up a project initially with a grant from Bowel Cancer UK, led on the NCRAS side by Stephen Hardy, Fiona MacDonald and Corinne Mallinson, and then taken over by the Clinvar, or with supplementary funding from this program grant from Cancer Research UK. So Clinvar was a 4.2 million Catalyst Award by Cancer Research UK to drive this whole process of variant interpretation. And as you can see from this very busy slide, an awful lot of bodies have become involved in this. And it's coordinated and led by Professor Claire Turnbull at the Institute of Cancer Research, who very kindly gave me the next few slides. The bioinformatics at the National Disease Registration Service is led by Francesco Santaniello and Ollie Tulloch, who've done the work which I'm about to show you. So basically what we've been able to do is gather genotype and phenotype data from all of the laboratories around England and Wales and pool them together for the purpose of this analysis. So here's some of the data that we'll be reporting back to Bowel Cancer UK. For example, on colorectal cancers in 2018, there were 35,851 colorectal cancers, of which 1.9% were subjected to a germline test for Lynch syndrome. Well, clearly a good deal less than we'd like, because we know that about 3% of them will be Lynch syndrome. And to find those 3%, we should be testing about 9%. Similarly, endometrial cancer is under-tested, though not quite to the same degree. And the urothelial cancers of the kidney, ureter and bladder, which are also features of Lynch syndrome, we have 10,000 and more of those each year. And in 2018, only 43 of them were subjected to germline testing. So clearly, at a national level, we need to improve our investigation of these patients to identify which are carrying pathogenic variants in the mismatch repair genes. We're also able now to access the laboratories to look at the immunohistochemistry and the MSI testing and feed that back into the system. And Stephen and Fiona and I will shortly be producing a paper bringing all of this information together to show the degree to which we are succeeding in fulfilling the nice guidance in the UK that all patients with colorectal and endometrial cancers should be checked for Lynch syndrome, because we're clearly still some way off that ideal. This is the total data acquisition that we've managed to connect together from the genomic laboratories and the Cancer Registration Service. And you can see that the blue columns, which are the breast cancer genes, have been the primary focus of interest here, with in December 2020 having reached over 100,000 investigations. This is a relatively small number compared to some of the diagnostic labs in the States. But remember, all of these patients have been selected because of a high Manchester score. That is to say, there's a greater than 10% probability of them having a BRCA1 or BRCA2 variant. And what Claire and her colleagues have done is taken the 22,594 probands where we have a white ethnicity to do some interesting work on whether we can use that to identify the likelihood of pathogenicity based on the so-called PS4, where we're looking at, because this is a total population study, we can look at the frequency with which variants are being identified in these breast cancer families and compare that to the other non-cancer data that we have available to us. And the work here is very much dependent on Kankut Kubuk and Subin Choi working with Claire. So the idea here is we're looking for an odds ratio. We're looking at the cases, how many of breast cancer patients, for example, develop with a variant are seen, or sorry, how many cases of a variant are seen among a population of breast cancer cases, and compare that to a control population to give an odds ratio. So in the first release of those 22,500 cases, Claire was able to identify 233 variants of interest, which would fit in with the ACMG category and worthy of investigation. And just to look at one of those, for example, here is the BRCA1 codon 213 to 11 T to G. And what we've got here in the breast cancer cases was 11 of those patients had this variant out of the 22,000. Whereas in our control population, from NOMAD and from non-cancer referrals, it was a much rarer observation. So that gives an odds ratio of 12.5, and according to our understanding of this, that looks like a strong evidence that this is a pathogenic variant. This is clearly a topic of great interest and debate, and the ideas are set out by the CanVig team in their recent publications. But I think this is showing us a direction of travel, that what we might see in the future is a pooling of data from all of our diagnostic laboratories through that anonymization system into the cancer registry, so that we can just systematically look at all variants that are being identified and using an artificial intelligence approach, start to pick out variants of interest, particularly when we start to get into the lower penetrance conditions. That obviously will be a way forward for the future. Before I end, I'd like to just go back to the hemoglobinopathies, because we began there with sickle cell disease. When we set up the BRCA challenge, our colleagues in the global Variome at UNESCO from around the world said, this is all very interesting, but actually breast cancer isn't our biggest problem. And so we agreed to also start a collaborative project on thalassemia. Now, this is to some extent a solved science, but there is still a great deal to be learned about the diagnosis and management of the different variants in the hemoglobinopathies. So these are the countries around Southeast Asia and their favorite variants in the beta globin and alpha globin genes. And if you look across at China and India, you see a slightly different pattern. Again, like sickle cell, these variants have been made common because of the prevalence of malaria in years gone by, or indeed still the case, which has made these patients more likely to survive, or these alleles to survive and prosper. So just to remind ourselves that when we are thinking in the high income countries about solving these problems, we also need to think about how we help our colleagues in low and middle income countries to try and solve these problems too. So it's not just a case of identifying variants of interest. We also need to be thinking about how we pool that knowledge so that we can produce highly efficient, low-cost diagnostics to roll out screening, as has been demonstrated in Cyprus, where they were spending 10% of their health budget on treating patients with thalassemia before introducing a national screening program. It's also worth remembering that we're still at the beginning of a long journey. We're still focusing on the high penetrance monogenic disorders. And the minute we start to get into more complex genetics and genomics, clearly we've got a very long way to go in understanding how to differentiate those variants from that are significant from those that are not. And I'd like to just show you one last piece of information from a little project that I've been involved with in Newcastle around the COVID crisis. As you will all be aware, there's been a lot of interest in looking for genetic variation underlying the variation in susceptibility to COVID-19. And one of my colleagues, David Langton, who has a special interest in rejection of joint replacements, has great interest in the HLA system. It was interesting that the evidence from the literature on genome-wide association studies is somewhat ambivalent as to whether HLA is involved, which is a little surprising, given the nature of the disease. So what we set up in Newcastle was to look at a relatively small number of people who had had full-blown severe COVID-19 requiring intensive care, and comparing that to asymptomatic or minimal symptomatic patients, or hospital staff who had COVID-19. And what came out of that analysis, which is shortly to appear in press, was quite interesting difference in DRB10401. This is a variant which is common as a factor in rheumatoid arthritis. And you can see that only 5.1% of the people with severe disease developed that, whereas 16.7% who were asymptomatic had that particular variant. And the reason that's also interesting is that when you look at the distribution of the frequency of that particular allele with latitude, what this graph is showing is that there is a significant correlation. The further away you get from the equator, the higher is the incidence of that particular allele. That might be a factor in understanding why we've had such a major problem in the UK of ethnic minority people developing the disease, because of course, if you are a person with a slightly higher susceptibility mixed in with people who are more likely to be resistant, then you're going to get a higher rate of asymptomatic viral carriage and transmission. And so that might be a factor on top of the other factors such as BMI, etc., that have influenced the high prevalence in certain communities. So just a final word, that the Human Variome Project does still exist, and it's now merged with the Human Genome Organisation alongside the Human Genome Variation Society, and we'll be hearing in this conference from my colleague Mark Greenblatt from HGVS. And so we've just in the last couple of months merged the three organisations together to hopefully continue to contribute to this important area of endeavour. So in summary, gathering and sharing knowledge of the clinical significance of genomic variants is a common good. And I think we should never lose sight of the need for us all to work towards making accessible information to all people on the planet that is of relevance to their healthcare. Most variation exists outside the white European ethnicity group, so we do particularly need to focus our attention on the low and middle income countries, where there is a wealth of information and knowledge to be gained, and who will benefit in return from us working with them to identify variants. National resources are needed to collect and curate variants of interest. We've been working now for decades to try and run this on a shoestring with different grants and so on. And I think we need to recognise that our healthcare systems need to build this into their system. And in particular, functional assays to try and sort out the importance of different variants should be resourced within healthcare systems, which I think is a case that we all have to try and make to our colleagues in government. Effective curation requires accurate and unbiased phenotypic data. And so it isn't sufficient to sequence and pool variants. We need to work with the clinical community to gather reliable phenotypic information and make that available, perhaps using the pseudonymization system that I've told you about today. Pseudonymization provides a path to systematic variant classification, certainly in healthcare systems where we have an integrated healthcare, and I hope that that can make a useful contribution to the field in the years to come. Thanks for your attention.